President, Vice President, members of Council, past President, fellows, members, distinguished guests, it's a real pleasure to welcome you to RCSI this evening to the occasion of the 100th Abraham Colles Lecture. Now, it gives me pleasure to invite the President of the Royal College of Surgeons in Ireland, Professor Deborah McNamara, to introduce tonight's lecture.
Past Presidents, RCSI, Vice President, Members of Council, Vice Chancellor, Deputy Vice Chancellor for Academic Affairs, Fellows, Members, Affiliate Members, distinguished colleagues and guests. The Abraham Colles Lecture Series was established in 1956 and is regarded as RCSI's most prestigious lecture, reserved for senior individuals who have achieved eminence in their field. It honours Abraham Colles, who was born in Kilkenny more than 250 years ago. He trained in Dublin, Edinburgh, and London, and he is regarded as one of Ireland's greatest 19th century surgeons. He was appointed dual professor of anatomy and surgery in RCSI in 1804, and he's said to be the person who, more than anyone else, made the profession of surgery respectable in Ireland. In itself, I think you'll agree, a noteworthy achievement. He's best known for the fracture of the distal radius that bears his name. What's perhaps less well known is that he revolutionized the teaching of anatomy by changing from the previous systems-based approach to one where anatomy was taught on a regional basis, focusing more on the relationships between structures that is so important to the practice of surgery. This year's Colles lecturer also has a unique perspective on the relationship between structures and surgery. Professor Fergal J. O'Brien is Professor of Bioengineering, Regenerative Medicine at the RCSI University of Medicine and Health Sciences, where he is also head of the Tissue Engineering Research Group and Deputy Vice Chancellor for Research and Innovation. Fergal achieved his primary degree from Trinity College Dublin in 1997, his PhD in 2001, and was a Fulbright Scholar. Since his faculty appointment to RCSI in 2003, he has published more than 300 peer-reviewed publications, filed 20 patents, and supervised more than 60 doctoral students, many of whom join us here this evening. He is co-founder of 3 spin-out companies, an editorial board member of 10 journals, and as of this month has received 4 European Research Council awards, including the prestigious €3 million ERC Advanced Grant, often considered the most prestigious research funding award in Europe. He has received numerous awards and fellowships from learned societies and is uniquely interdisciplinary, spanning the fields of engineering, bioengineering, medicine, and surgery. In 2018, he was elected member of the Royal Irish Academy, the highest academic honour in Ireland, and in 2023 received the Marshall R. Urist MD Award from the Orthopaedic Research Society. 100 years on from the first Colles lecture, Professor O'Brien's groundbreaking work in bioengineering and regenerative medicine reframes our understanding of anatomy for another generation. And I can think of no one more deserving of the honour of delivering the 100th Colles lecture than this year's lecturer, Professor Fergal J. O'Brien.
So thank you, Professor McNamara, and I'm just trying to keep roughly to time. So it really is a true honour, and to be quite honest, I'm so incredibly humbled to have the opportunity to present this lecture here today. And my great colleague Kieran Ryan has just reminded me that it's the 100th Colles lecture, so there's nothing at all to be nervous about. So thank you, Kieran. I'm going to take you really through a journey of tissue engineering in RCSI, and just to put some pictures maybe to what Professor McNamara has just talked about. So Colles was one of our early graduates in medicine in 1795. He subsequently became president, professor of anatomy and physiology, and professor of surgery. And what he is best known for is the fracture of the distal radius, which gives this standard dinner form deformity. And I guess for someone like me with a lifelong interest in musculoskeletal research, it even resonates more to be giving the Colles lecture here today. I am based in the Department of Anatomy and Regenerative Medicine, And I, as an anatomy teacher, and I'm on senior management now, so I don't do as much teaching anymore, sorry, Clive. But even to this day, we use many of the same principles, particularly on teaching clinically relevant anatomy. And at all times, remember that it's healthcare professionals we're educating and not anatomists. And if you go into our anatomy room, you'll see on a board up on the wall where Colles is there as the 6th professor of anatomy and physiology. And our current professor of anatomy and physiology, or current professor of anatomy, is Professor Clive Lee, and more about him momentarily. But I guess one of the biggest honors that RCSI ever distinguished upon me up until now was when I became professor, the first professor of bioengineering and regenerative medicine back in 2013. And I got my own name up on that board in the anatomy room. And I guess, what I want to talk about in the next couple of minutes is how that happened. How did this engineer suddenly end up teaching human anatomy, and indeed now coming to the stage where I'm giving this lecture? And for me, I had a very fortuitous encounter back in 1996 when this man on the right, Professor David Taylor, introduced me to Professor Clive Lee, who had just returned from a Fulbright scholarship in Harvard. And this led to me carrying out a final year project here in RCSI. First time I ever set foot on the campus was in 1996. That led into a PhD, and building on some of Clive's own work, where he had been using a fluorochrome labeling technique to look at cracks in bone, we developed a mechanism to monitor how cracks grow and how this leads to increased bone fragility. With the support of Clive, I applied for a Fulbright scholarship, and this is an actual slide from in the early noughties. So this is these lovely, tremendous blue background. That's how we presented things back those. I'm not sure our marketing department would like it now. But that brought me to Boston, and anyone who's here earlier will have seen Professor Ellen Roach's talk, and Ellen works in that same environment now, where I had the privilege to work in for a couple of years. And I worked with a material scientist, Lorna Gibson, who was working in the area of cellular solids, and a mechanical engineer, Ioannis Yanas, in tandem with Harvard Medical School. And I guess all throughout my career, I've had that privilege of working between engineering and medicine. And I think the innovation session commented on earlier on, but really that's the way that real disruptive science and research takes place. It's when clinicians, engineers, and scientists come together. It was a really interesting time to be in Boston, and because the field of tissue engineering was just beginning to develop. And this is a paper from Science in 1993, authored by Bob Langer and Joseph Acanti, where they defined this emerging field of tissue engineering, how the principles of biology and engineering come together to develop substitutes to repair damaged tissues. There is actually a Colles lecture link here as well, because Bob Langer, who would be seen as really the father of modern drug delivery techniques, his work has given rise to dozens and dozens of spin-out companies like Moderna, who people will be familiar with. His mentor was Judith Folkman, who discovered angiogenesis markers and really developed a huge amount of knowledge of angiogenesis. And Folkman was a previous Colles lecturer. So for someone like me, following in his footsteps is such an honor. Joseph Acanti was also a previous Colles lecturer, and even Joseph's brother Chuck, Charles, also gave the Colles lecture. So while I might be the first engineer giving this lecture, I'm not the first person just talking about the field of tissue engineering. So a little bit for people who might be familiar, what is tissue engineering? Well, if you consider at a fundamental level what our organs and tissues are made up of, generally you consider it as a three-dimensional framework of natural polymers like collagens and various types of biological molecules. And what we're trying to replicate in tissue engineering is the basis of an ideal implant for tissue engineering is to replicate that matrix. So what you see in that image is a porous collagen glycosaminoglycan, what we call a scaffold. So it's essentially an implant that it goes into the body, recreates the tissue, and cells then begin to behave normally. They understand the environment that they're back into. So I worked in Yanni Yanas's lab, and the interesting thing is, if you look at the years of those patents back that Yanas developed, and Yanas developed really the very first scaffold for tissue engineering, a collagen glycosaminoglycan copolymer, We never called it a scaffold. We called it a matrix. And the term that had been coined in the late 70s and early 80s was artificial skin, because what that material did, and what you see in the image on the left, is what it looks like physically if you were a surgeon, where you will take that sheet, cut it to fit the size of the defect. And on the right-hand side, you can see some early clinical trials work where this is a boy with third-degree burns. His lower abdomen has been treated with a sheet of the scaffold, and his upper abdomen has been left untreated. And you can see how it inhibits scar formation. From Yanas's lab, spun out a company called Integra Life Sciences, and I'll come to them later. But that technology, Integra marketed that, and that's been used in lots and lots of patients, particularly in the area of wound regeneration subsequently. But I'll come back to that later on in the talk. So when I was recruited to work on bone. But actually, there was some issues with the scaffold, so I didn't actually spend much time working on bone. Instead, I worked on a lot of fundamental scaffold development research. And as an engineer, I always have to have some equations and a presentation, but I promise I won't go through them in any great detail. But we published a couple of important papers during my time in Boston, and those papers have become quite well cited over the years. And they became the basis of my first significant grant when I came back to Ireland. So Clive Lee, again, as a mentor, had the vision to employ an engineer and get an engineer to teach anatomy to medical students. So my faculty appointment was here in the Department of Anatomy. The department is now full of engineers, maybe too many, we think some days perhaps, but it's shown the evolution of that. When I came back, I was fortunate enough to receive President of Ireland Young Researcher Award, and that led to me establishing a laboratory for tissue engineering. And that enthusiastic young person in the middle, that's Bill Harris, who was the founding Director General of SFI, obviously with President McAleese. But SFI had just been established. It was a really exciting time to be in Ireland. And in parallel with that, the government had found the programme for research in third-level institutions. So for the first time ever, funding was going into the universities. And this led to the establishment of the Trinity Centre for Bioengineering, which throughout the years has had a significant RCSI part in terms of leadership. I'm delighted to see Professor Patrick Prendergast here, who was the founding director of TCBE. It led to the development of a national network in the area of biomedical engineering. And over the subsequent 25 years, really what has happened is bioengineering has gone from strength to strength. And I don't think it would be false modesty to say that Irish biomedical engineering is not ranked in the top areas in the world. It's become a global hub, but that's happened in parallel with the growth of the medical device industry. And in 2018, Danny Kelly and I co-chaired the World Congress in Biomechanics. And as part of that, we authored a paper, Danny, myself, and Paddy, just to describe how the field has grown. So for anyone who's interested in the evolution of a field, I think it's worth reading. So that's what the group looked like back in 2005. You might recognize a couple of people. Oren Kennedy and Orla Brennan are faculty members here in RCSI today still. We've grown a bit subsequently over those years. And really what we do, to talk a little bit on the science, what we do is use these biomimetic implants, these biomimetic scaffolds, both for tissue repair, but increasingly we use them as model systems, as templates to understand more about disease and use them for drug screening. And I'm going to talk in remembrance of Colles a lot about musculoskeletal research, but we've now got programs of research going on in all sorts of tissues within the body. But regardless of what tissue we work in, the same fundamental facets remain the same. We look at the tissue, we look at the matrix biology, we develop the material, the scaffold, to replicate that biology. And then we use different types of processes to try and turn that into an implant that will be receptive to cells when it goes back into the body. So with the first cycle of PhD students that come in back there, you'll see Michael Keogh on the left-hand side, Ciara Murphy, who's now faculty in RCSI, Matthew Haw, who's here today, I think, as well, too. So these talented researchers became the first group, the first cohort that began to move us, I guess, onto the next stage. And our first sort of success story was in the tissue that I was supposed to have worked in Boston, excuse me, and that was in bone. And I talk about biomimetic. At a simple level, bone consists of two things: collagen, which gives us its tensile strength and its biological functionality, and a ceramic hydroxyapatite. For years, people have tried to use collagen scaffolds or hydroxyapatite scaffolds. And I guess what we tried to do was to combine the two together. I got funding from Enterprise Ireland that allowed me to recruit this man on the left, John Gleeson. And we took that technology to a lot of different types of characterization methods. We tested it in the first animal model. That was in a rabbit model, and fittingly, it was in a rabbit radius, to go back to the Colles link. Those surgeries were carried out by an orthopedic trainee, Frank Lyons. And I guess what I should say too is that throughout our development as the group has grown, we've tried to keep clinical trainees involved in our research to carry out the surgeries when we do these types of studies. Our first clinical study was actually in a horse, and where the material was used to treat a racehorse's mandible. The veterinary school in UCD came to us with this case. The prognosis for the horse wasn't particularly good. Unfortunately, the material began to work well. And I just, if I diverge for a second, because I see the name of the product, John and I, with our scientific hats on, thought that we should call it hydroxycol, taking into mind both components of the materials we were using. I remember one of my orthopedic colleagues telling me that was the wrong choice many years later, that we should have made it sound like something like a golf club or a golf driver if we really wanted to sell that to orthopedic surgeons. But in parallel with that, we started to, and Aoife Gallagher, who was working in our technology transfer office at the time, is now RCSI head of innovation. We spun out a company. That company raised investment. It got to a stage where we got regulatory approval for it. And indeed, the very first in-man human case study that was carried out with that scaffold was carried out by Dylan Murray in the Mater. I think Dylan, I just met him on the way and is actually here today. In parallel with that, and I guess the reason why we spun out a company rather than license the technology to a multinational, which might have made it an easier route to the market, was that in parallel, we had developed a scaffold for cartilage repair. And as people will be aware, cartilage is a completely different beast in terms of a tissue that you try to heal because of the fact it's got such a limited regenerative capacity. So John and I wrote a grant. We recruited this smart postdoc, Tanya Livingston, and we developed this multi-layered collagen-based scaffold for osteochondral repair. And this scaffold was designed to replicate the natural tissue that you find within the articular joint. So multi-layered structure with a gradient in stiffness and in composition. And really the principle behind that is that when the scaffold will be implanted into the defect, it would soak up the body's own cells, and then the composition of the material would direct functional repair of the joint. And then the story was pretty similar to bone. We tested that initially in rabbits. We subsequently tested it in goats in partnership with Professor Peter Brahma in the UCD Veterinary School. And our first clinical case was again in a horse. And this time it was a show-jumping horse who presented with osteochondritis dissecans, which is quite common in thoroughbred horses. And it worked effectively. And that got a lot of media attention, both nationally and internationally. And what I'd love to say that the reason it got the media attention was because of the science. I think it was possibly because of the horse's name, because the horse was called Beyonce. So you had international websites with headings saying things like Irish scientists cured Beyonce's sore knee. And I think when people clicked on that, they might have found something different than they were expecting. One of the great things working in RCSI is the engagement with clinicians. This point has come up time and time again. How do we innovate? How do we take science forward? Well, really, you need multidisciplinary teams. And I've been fortunate enough throughout my career to work with Professor John O'Byrne, who's the Abraham Colles, fittingly, Professor of Orthopedic Surgery in RCSI. So John and Cathal Moran worked with us to develop a clinical trial, and this is unpublished data, which we only finished a couple of years ago, where we showed that that scaffold could work effectively in treating knee lesions, focal knee lesions in people. And I see Tanya in the audience. Tanya, we are going to publish that paper this year. That's definitely happening. But I guess that's the first part of the talk that I wanted to talk about, how we develop scaffolds and how we focused on translating. What I will say is that those scaffolds will only be effective at treating certain size small defects. So with a view to scaling up towards much bigger defects and to more traumatic injuries, over the last decade, we've really been focusing on using them as delivery vehicles for different types of therapeutics. So the same scaffolds as before, except now they have this capacity to deliver therapeutics to the site of where they're needed. And again, I think in my career, I've been very fortunate to be in the right place at the right time. And that was the case back when we started these studies, because these three people, Gary Duffy, Sarlianne Cryan, and Helena Kelly, were all based here in RCSI, and they really gave us a skill set to start looking at drug delivery systems. So as an exemplar, and sticking with bone, if you look at the image on the right and you look in closely, you can see microparticles, same scaffold as before, but it's embedded with microparticles which are controlling the release of therapeutics. So we showed that this scaffold-based system, this is a dual release system with two therapeutics being released at two different rates. We could accelerate the bone repair process. We also applied that platform in the area of infection, and particularly in the area of osteomyelitis. So we've done a lot of work with osteomyelitis over the year in partnership with Professor Steve Kerrigan in particular. So we used that same platform for the dual release of an antibiotic or different types of antibiotics. And the dual responsiveness gives us the opportunity to deliver one drug, one antibiotic to knock out the initial infection, and the secondary release then to knock out any subsequent bacteria that might begin to grow. And we licensed that technology to a company in the UK, and actually that patent was just granted many years later, back in November. And I guess one thing I wanted to show for maybe earlier career people, the paper that came from all that body of work was just published a couple of weeks ago. And the message I would give is that when you have an idea, please talk to Aoife, Derek, and the team to see when it might be worth protecting the IP before publishing the paper. Anytime we have patented, we have typically followed on with a paper subsequently, and it may be years later, but the story remains true. In some ways, the work that we've become best known for is using these scaffolds to deliver gene therapeutics. So everyone, thanks to COVID, is now aware of things like RNA vaccines and of gene therapeutics. And for a long time, it had seemed that this whole field of gene therapy was dead in the water. And indeed, the first grants I ever wrote were rejected because I was told that the field was dead in the water. But fortunately, the European Research Council gambled on us. And what we now have here, same scaffolds as before, same porous architecture as previously, but we have nanoparticles embedded within the scaffold matrix. And these nanoparticles are complexed with different types of genetic cargoes. So Gary Duffy and I recruited a new team of PhD students to focus on this area of delivery. We also recruited a really talented postdoc, that woman in the middle, which was Caroline Curtin, and also Erica Tierney, who's a PhD student, who I'm delighted to see her in the audience here all of a sudden today. What that's given us over the last decade or so is a full library of different types of nanoparticles. And we can now use these particles to release different types of genetic cargoes at different speeds, at different rates, depending on the tissue of interest. But again, our first choice, or our first target was in bone. And this was really seminal work by Caroline Curtin, who's now faculty in RCSI, assisted by a PhD student, Grania Caniff. We showed that the same hydroxyapatite particles that we had used to make the scaffolds, if we used them in smaller form, we could deliver genetic cargoes to bone defects. And that kind of concept worked really well. And it opened the door for us to be able to deliver other types of cargoes, such as RNA therapeutics. This is an example of a dual delivery of microRNAs, for example. We've been delivering microRNAs in combination with molecules such as antimicrobial particles. And in tandem with Sally Ann and Andreas Heiss's group, we've even been delivering messenger RNA-based therapeutics from these scaffolds. And I guess what that did was it's opened the door for us to use the same use particular scaffolds. The advantage of the scaffold is it gives localized delivery of the therapeutic. And it's opened up a whole wide range of different types of projects from which we can work with. As an example, in cartilage, we got more funding from the European Research Council. We started using those scaffold systems to deliver targets, deliver molecules to enhance chondrogenesis cartilage deformation, but also to silence inflammation. And Tom Hodgkinson was a postdoc we recruited who's also faculty in RCSI, is now developing his own independent group in this space. And really, the final area I just wanted to talk about briefly is how we moved from musculoskeletal into different indications. Really, that came about because as the group grew, we, I suppose, began to develop a name for some of the work we were doing. But much of it came about because clinicians would come to me and say that they had a particular problem. And I'll give an example of that. We had developed a material for corneal repair in partnership with Conor Murphy here in RCSI. And an ENT surgeon, Alec Blaney, was in my office and he picked up the material and he said, have you ever thought about using this for tympanic membrane repair? So that kind of interaction, again, between clinicians giving us problems we hadn't even thought about and us having the engineering capability to come up with solutions has been hugely beneficial. So, for example, we combined elastin and collagen materials to look at vascular grafting. We used that same material, that tubular material in the middle, and if you look closely on the inside of it, you can see a film. That film formed a functional endothelium. With Sally Ann Cryan, we used it we recruited a PhD student, Keane O'Leary, and subsequent students, and to look at airway regeneration, where what was an endothelium became an epithelium. Claire Brom was a PhD student who worked on heart valve. And indeed, Claire is now faculty in TUD, and I see her here today as well too, and is now leading her own group on that and sticking with the heart valve theme, and it was a major focus of the previous session. Claire Conway, who we recruited into the anatomy department, is doing a ton of work in computational modeling in this cardiac area. Michael Walsh in Limerick has always told me that there was some low-hanging fruit in the area of urology, and I'm delighted to see Michael here. So we've been working with Niall Davis, Michael and Owen Canan in the area of urology. We've always been interested in cancer because it goes back to what I said about the materials being capable of being used as test beds. They work well to grow tissues, so they work well to model tissues as well too. And now Olga Paskareva and Caroline Curtin are doing a lot of work in this space themselves. And I guess coming full circle to the project I started in MIT when I was supposed to work on bone, the challenge Yanni and Ass always told me that the skin problem was ripe for more development and never felt the Integra scaffold was really optimized to the level that it could be. So we've got a very active program of research in the area of wound healing, and this brings in Shane Brown's expertise, where the scaffold is slightly more complex because we've introduced an epidermal layer on the scaffold, which is also antimicrobial to kill bugs. And Michael Keogh, our former PhD student, is now leading a satellite lab of the group in RCSI Bahrain, where he's been using these gene-activated scaffold systems in the area of diabetes treatment, which is hugely prominent in the Western world, but also very prominent in the Middle East. And we've just started a project in the area of epidermolysis bullosa, funded by the HRB and Deborah. Again, just showing the multifunctionality of the same platform. You'll see a couple of slides with the amber logo on it. What AMBER, the Advanced Materials Bioengineering Research Centre, has done for us is it's allowed us to really partner with industry. And when you're working in the area of biomedical engineering and in the medtech space, the only way you can take science out of university labs for the benefit of patients is to work with industry, either through spin-out creation or through partnering with multinational companies. And I'll give an example of one industry partner. It's the same company that I mentioned at the start, Integra Life Sciences. We've been working with Integra for many years now. And what we've been doing is they had a market-leading nerve regeneration product, which was basically an empty collagen tube. And what we've been doing is filling that tube with a matrix that can help nerves to grow better. And Integra have just funded, I think, the 4th project in our lab to look at ways to even treat bigger defects. But our work with Integra on a peripheral nerve repair led us into the last area I just want to mention briefly, and that's our work in spinal cord injury. And this is another Amber Centre partnership, but rather than working with an industry partner, we've been working with the IRFU Charitable Trust. So this came around from conversations with Cliff Byrne and John Quinlan, and we've taken that forward. Now, as people will be aware, spinal cord injury really is the holy grail of regenerative medicine. It's a hugely complex condition, and every patient's needs are completely different. So for a complex condition, you need a complex solution. So very fortunate that I recruited a really great neuroscientist, Adrian Dervin, and we've built a team over the years to try and tackle this. So we've started with our multifunctional scaffold again. We've been using that scaffold to deliver gene therapeutics again, both to enhance nerve growth, but also to reduce glial scarring, which is a major problem and a major risk factor or a major issue with why nerves can't grow. But I think in some ways, and maybe it's coming back to the full circle on the engineering side, the area that I think we've been making some really interesting progress is using these materials to deliver electrical stimuli. So as well as being biologically functional, and as well as being able to deliver genetic cargoes, these materials now can carry electrical stimuli. And that's been really interesting. And it's come about a lot because of our Amber Centre partnership, where we can collaborate with material scientists to try and drive this forward. And that has also been really beneficial in that it's brought proper public and patient involvement into our research programs. And I can't really talk enough about how important it is to develop these programs in research programs in a medical university like RCSI. And we're very fortunate to have people like Michelle Flood and Niamh Dillon who are helping this to happen. I've talked about how we took the same kind of platform and moved it into different applications. One of the reasons when I first started here back in 1996, and I look at how different the institution now is from back then, huge amount of that is down to the growth in our investigator base and how our kind of research culture has grown over time. So things like the Strategic Academic Recruitment Program have allowed us to grow, but have also allowed the institution to grow. And I have to say, for somebody interested in innovation, it's such a privilege now to see the pipeline of companies that's growing both from the tissue engineering group, but also from the university as a whole. And here's some examples that are just beginning to come back. It's a real privilege to work with people like John Leeson again in a new indication. And that was a tympanic membrane idea that came from Alec Blady. And people like Helena Kelly, we've just heard from Amir Hamid. These are companies who are already on the journey and hopefully will begin to move forward. And the reason why I sort of ended up eventually as Deputy Vice Chancellor and ended up in the research office was Ray Stallings inviting me to come in. He was Director of Research back in 2012 to assemble a team to grow innovation. And in the following 10 years, it has been such a privilege to work with people like Aoife Gallagher, Seamus Brown, and our professional team in the ORI to help to grow research and grow innovation metrics. That's led to, and Colette Power and Estates knows all about the challenges with research funding growing, but it's led to a huge growth in research activity in the institution. It's led to a huge growth in postgraduate registrations, and huge credit to Darren O'Connor and his team at the School of Postgraduate Studies for moving in that. But the key enabler is the growth of investigators. And I've talked about how things like the STAR program have helped that to develop. But the real key has been how we've developed more collaborative relationships with our clinical partners. And I think it's come up time and time today about how important this is. And I think Ireland is challenging. It's a challenging environment to start clinical trials. We're slowly making progress, but it's something we've got such an opportunity to do. And I look at our RCSI Clinical Research Centre is now beginning to develop a plan to grow and grow. And I think the National Surgical Research Support Centre also will be a real enabler of this as we go forward. And finally, I guess, while it's good to publish papers and it's good to create PhD students and it's good to create companies, it's absolutely critical for the next generation that we tell people and we tell the public about what we do. And I think our new public engagement space that's coming here on St. Stephen's Green in a few months' time is going to be a real key enabler to allow us to do that. And just to finish up with, and I promise I'm not going to name all these people out, but I really consider this, I'm so honored to receive to give this lecture, but I really see it as an accolade for all the wonderful people who've worked in my group over the past 20 plus years. And I think, you know, the things that have excited me most are seeing trainees' careers going from strength to strength. And it's such a privilege that many of the former trainees are now collaborators and indeed good friends. You can't do this kind of work in isolation. The field we work in is incredibly interdisciplinary. We need funding. We've been fortunate we've gone through different funding cycles, but generally we've been fortunate that Ireland began to invest in research, and it's so important that this continues to do so. But to do real world-class research, you have to look internationally. And we've many collaborators, both at home and abroad, who've helped us grow over the years. And my last slide, and the person who I need to thank most, is my wife, Orla, who has supported these adventures and this work, particularly when I'm away on RCSI business or telling the world about what we actually do. And Orla's mum Maura is here, and my twins, Idan and Lana, I see for this lecture have managed to stay awake. So, go raibh míle maith agaibh go léir.
I don't know how you follow that, but all I can say is my sincere congratulations to Professor Fergal O'Brien for an outstanding lecture. I know our president has outlined Fergal's contribution, both institutionally to RCSI, nationally to organisations such as AMBER and the National Surgical Research Centre, and indeed internationally to his work with the European Research Council. And I think his lecture has captured that work very well. And my sincere congratulations and indeed thanks to him on behalf of the so many people he's supported in their careers. Can I also, at this point, give my sincere thanks and congratulations to our president, Professor Deborah McNamara. The president very much leads the Charter Week, and this week has really reflected her priorities as president for RCSI, creating an inclusive college for all our fellows, members and affiliated members, of encouraging our younger fellows and early stage consultants to participate on the national stage here at RCSI, a college that's actively engaged in the real challenges facing the Irish Health Service, a college that is alert to the future advances in technology and digital health, ensuring that our fellows are ready to embrace these opportunities. In my view, the meetings over the last week have very much reflected these important priorities. My thanks also to our sponsors, the organisers of the Robotic Village, the organising committee, our excellent speakers, session chairs, and finally our professional services teams who make it all seem so smooth and effortless, our conference and events team, media services, portering, estates and catering teams. Colleagues, that concludes the formal part of our charter meetings. I'd like to remind you that the Irish Surgical Training Group have their annual meeting tomorrow morning here at RCSI, and the Bosco O'Mahony lecture, which headlines that meeting, will be delivered by my colleague, Miss Dilly Little, consultant urologist and transplant surgeon at Beaumont Hospital. Can I thank you all for attending and your support and ask you to be upstanding for the outward procession.